Good afternoon, and thank you all for attending today's journal club that's sponsored by the American Thoracic Society, Allergy, Immunology, and Inflammation and and Inflammation Assembly. My name is Amit Bery, and I'm an instructor of medicine at Washington University School of Medicine in St. Louis, Missouri. I will be moderating today's webinar. Today, I'm very excited to discuss the paper entitled Metabolic Rewiring Promotes Anti-Inflammatory Effects of Glucocorticoids, published in Nature. We are fortunate to be joined today by the first author, Jean-Philippe Auger. He's a junior group leader in the Medical Department of Rheumatology and Clinical Immunology under Director Professor Dr. Gerhard Kronke at Charité University of Medicine in Berlin. Their group investigates basal molecular regulatory and control mechanisms governing innate and adaptive immune responses with a specific focus on the role of distinct immune cell types during homeostasis and inflammation in autoimmune disease, along with assessing the impact of metabolic processes on immune cell function, also known as immunometabolism. We are also fortunate to have two expert discussants joining us today. First, Dr. Barbara Balestrieri, who is an assistant professor of medicine at Harvard Medical School and an associate immunologist in the Division of Allergy and Clinical Immunology at Brigham and Women's Hospital. Her work focuses on understanding the pathogenesis of asthma and lung diseases. She has previously highlighted the critical role of macrophages in both initiating and sustaining the inflammatory response to allergens, and more recently, has explored the interactions between macrophages and various other immune cells. Next, Dr. Vidula Vachharajani, who is a professor of medicine at the Cleveland Clinic Lerner College of Medicine and is director of translational critical care medicine at the Integrated Hospital Care Institute. She's also co-director of the Center for Sepsis-Driven Immunodeficiency at the Cleveland Clinic in Cleveland, Ohio. Dr. Vachharajani is a critical care physician scientist who studies sepsis. Specifically, she studies immune metabolic responses in sepsis and septic shock. Currently, her laboratory is studying the role of a nutrient sensor protein, sirtuins, in suppressing immune response during late sepsis. In addition, she also studies how alcohol use disorder dysregulates glycolysis and phagocytosis in macrophages. If you have any questions for the presenter or the discussants at any time during the webinar, you can enter them in the chat. There'll be a moment at the end where you'll be allowed to unmute yourself at the end of the presentation for a Q&A session. And now I will turn things over to Dr. Auger.
So I'd like to start off by thanking Dr. Bery for the kind invitation and to share this work with you. So this was work that I did during my postdoctoral fellowship, and I'm very excited to share what we discovered. So for those of you who are a little less familiar, glucocorticoids are one of the most important therapies in medicine. They're used to treat a wide variety of diseases, going from infectious to autoimmune and immune-mediated. And one of their characteristics is that they actually affect pretty much every organ in the body, including the immune system. And they're best known for their anti-inflammatory effects, so therefore they do target the immune system readily. And the current paradigm regarding the mechanism of action of glucocorticoids is pretty straightforward in that basically the glucocorticoids manage to cross the cell membrane without a transporter, and they will bind the receptor, which is located in the cytoplasm. So ligation of the glucocorticoids to the receptor leads to translocation of this ligated receptor to the nucleus, where it will act as a nuclear transcription factor, and it will bind to GR response elements in various genes, and this leads to their transcriptional activation in a mechanism called transactivation. However, the anti-inflammatory effects of glucocorticoids are actually less well known. So the current paradigm dictates that the receptor can also bind to other genes that do not have these GC response elements and turn down their transcription, and that this would be responsible for the anti-inflammatory effects. However, these mechanisms don't fully explain how the glucocorticoids have their anti-inflammatory effects, given that a lot of genes that are affected don't have these response elements. And this brings us to basically macrophages. So our lab focuses on many different immune cells, and one of our favorites are macrophages. And macrophages are central to inflammation, given that they're in every single tissue, and they're a hub for the production of inflammation but they're also important for inflammatory resolution and tissue repair. And this makes macrophages an important target of glucocorticoids, seeing as they're everywhere and that they're central to inflammation. And what we first wanted to do was basically just to have a general look at how glucocorticoids affect macrophages. So we used bone marrow-derived macrophages as our model because these are basically just a model in vitro culture that readily mimics macrophages. And we treated the macrophages with either glucocorticoids. So we used dexamethasone, which is one of the most potent glucocorticoids, or LPS, which lipopolysaccharide, which is basically a very strong activator and induces a pro-inflammatory phenotype in macrophages. And the cells were also co-treated. And we performed just a basic bulk RNA-seq experiment at both an early time points of 4 hours and a later time point, 24 hours. And what we were quite surprised to see is that at early time points, actually, glucocorticoids don't seem to affect overall gene expression as strongly as at later time points. So the current paradigm dictates that this repression of inflammatory genes happens very quickly. But if we look at, for example, at 4 hours, we actually see that a lot of the genes are quite similar in glucocorticoid-treated pro-inflammatory macrophages as without treatment. However, at later time points, such as 24 hours, we can see that there's a great difference, and that the glucocorticoid-treated pro-inflammatory macrophages much more resemble the control or glucocorticoid treatment alone. And indeed, a great number of genes are both up and down regulated following glucocorticoid treatment. Some of them not very surprisingly, such as IL-1 beta and NOS2, for example, very classical pro-inflammatory genes, while others, such as CD163, were actually upregulated. And we just then looked at different pathways using pathway analyses. And not surprisingly, inflammation was one of the pathways that was most regulated. But what did surprise us is that different metabolic pathways popped up as being strongly regulated, including oxidative phosphorylation, which is key to mitochondrial metabolism, as well as citrate cycle and pyruvate. So this seemed to suggest that the glucocorticoids differentially modulate metabolic pathway expression in pro-inflammatory macrophages. And this got us very excited because of the concept of immunometabolism. So this concept has emerged in the last 15, 20 years and has really demonstrated that there's a mutual influence of cellular metabolism and the immune response, particularly glycolysis and oxidative phosphorylation involving the TCA cycle. And this is the case in macrophages where pro-inflammatory macrophages usually express high levels of glycolysis, which they used for many of their pro-inflammatory functions, while anti-inflammatory tissue repairing macrophages generally rely a lot more on oxidative phosphorylation. So this kind of led us to question if this metabolic reprogramming that seems to occur in glucocorticoid-treated macrophages might be one of the mechanisms related to their anti-inflammatory action. That's to say, does the glucocorticoid receptor upon ligation somehow affect mitochondria and mitochondrial metabolism, which is key to cellular metabolism? And is this related to the anti-inflammatory effects that glucocorticoids have been known for over the last 60 years and the reason why they're therapeutically used in medicine? So to have a better understanding of this, we performed some basic seahorse extracellular analyses. So we used a glycostress assay and a mitostress assay, which basically the readouts are indicative of glycolysis and oxidative phosphorylation, respectively. And what we were able to observe is that upon LPS treatment, macrophages induce glycolysis and basically have no oxidative phosphorylation, which is a very well-known phenomenon. However, what surprised us is that upon glucocorticoid treatment, we were able to observe basically a reversal in this phenotype. So a decrease in glycolysis and an increase in oxidative phosphorylation, suggesting that the glucocorticoids are rewiring the cellular metabolism during inflammation. And what's important to note is that glucocorticoid treatment alone in steady state doesn't seem to affect metabolism. So there really is this effect of rewiring only in the case of inflammation. We also performed, so if we just basically use an energy map, we can very nicely see that glucocorticoids alone don't seem to have any effect. LPS induces a nice glycolytic energetic state, while on the other hand, glucocorticoid treatment really brings the cells towards a purely energetic state, suggesting that they're using a lot of oxidative phosphorylation in mitochondrial functions. And we were also able to see this using mitotracker dyes and flow cytometry. So the cells were stained with mitotracker green, which is an indicator of mitochondrial size or mass, and mitotracker red, which is an indication of mitochondrial potential and fitness. And what we were able to observe is that LPS decreases mitochondrial fitness or functionality, if you want, while glucocorticoids seem to be reversing this phenotype and rescuing the mitochondrial functionality during inflammation. And this, combined with this rewiring of the mitochondrial metabolism, really indicated that glucocorticoids are somehow affecting especially the mitochondria, but also cellular metabolism on a general level. And one of the important questions was basically, are these effects due to simply the receptor translocating to the nucleus and acting as a transcription factor, which is the known mechanism? And we were lucky to have collaborators at the University of Ulm here in Germany who had mice where the glucocorticoid receptor is expressed, but basically it lacks this nuclear localization signal. So without this localization signal, the receptor can't translocate to the nucleus. So it is still functional, but it can't exert any genomic functions. And we repeated basically these experiments. And what we were surprised to see is that even without this translocation to the nucleus, this effect of the glucocorticoids on metabolic reprogramming was similar. So basically, the glucocorticoids were able to rewire the cell into an oxidative metabolic state, and the mitochondrial fitness did remain sustained, suggesting that this metabolic reprogramming is non-genomic. And this was kind of a big leap for us, because everyone has always said that the glucocorticoid receptor can only exert its functions via genomic effects. So this opened up kind of the whole study, and it exploded from there, and we were very excited to see where it went. So we really wanted to look a bit more at the TCA cycle, which is central to cellular metabolism. And the TCA cycle is a series of reactions. And what we performed was flux analysis using 13 C-labeled glucose. We fed the cells, and then we looked at basically accumulation of glucose into the different TCA cycle metabolites. And what we're able to observe is that during LPS treatment, the only metabolite that seems to accumulate glucose is lactate. While on the other hand, glucocorticoid treatment was basically leading to an accumulation of glucose into all of the TCA cycle metabolites, suggesting that unlike during LPS, where the TCA cycle is known to basically be blocked, non-functional, or very little functional, glucocorticoids seem to induce a cycling of the TCA cycle, and therefore an accumulation of glucose into the different metabolites. And this brought us to a further point, because what's well known in the literature is that LPS and inflammation in general often leads to a block of the PDH, so the pyruvate dehydrogenase, which is the enzyme key to converting pyruvate into acetyl-CoA. And since this happens, basically in the case of inflammation, pyruvate will then be transformed into lactate. So we wanted to look at lactate and acetyl-CoA, which is the downstream product of PDH from pyruvate. And we were able to observe that LPS basically decreases acetyl-CoA production, while glucocorticoids seem to be increasing it. And conversely, LPS induces lactate production, which glucocorticoids seem to reverse. So this seemed to really suggest that glucocorticoids are affecting the PDH activity in some way. And using some further experiments, so what we wanted to have a look at was basically if uptake of pyruvate or glutamine, which are two of the sources of intermediates for the TCA cycle, had an effect. So we blocked glutamine uptake, and we were able to see that when blocking glutamine uptake, there wasn't really an effect on this metabolic rewiring of glucocorticoids. However, if we blocked pyruvate uptake, we're able to see that the cells basically maintained a very glycolytic phenotype similar to LPS alone. And this really suggested to us that pyruvate uptake into the mitochondria was key, and probably due to this PDH. And by using microscopy and co-immunoprecipitation, we were able to observe that the PDH does seem to co-localize with or at least proximately, to the glucocorticoid receptor. And this only seems to occur when there is glucocorticoid present. So in the absence of glucocorticoids, they don't seem to co-localize, but when there is, then they do. And moreover, which was quite interesting, is that the PDH activity does decrease with LPS treatment. So at first it will increase, and this is just due to LPS first inducing oxidative phosphorylation, and then there's this block. So PDH activity decreases. However, the PDH activity was sustained during glucocorticoid treatment. So it really did seem to suggest that glucocorticoids are somehow rescuing the PDH activity in pro-inflammatory macrophages. And if the PDH activity is rescued, what we would expect is that pyruvate can then be converted into acetyl-CoA, and with the TCA cycle functioning, basically there would be production of energy and a more return to anti-inflammatory, or if you want, a pro-resolving phenotype. So these results were all very nice, and we see that glucocorticoids rewire cellular metabolism in macrophages. But how does this relate to the anti-inflammatory effects of glucocorticoids? So that was what we then wanted to investigate, because this could have just been two completely different mechanisms. So basically, glucocorticoids are affecting mitochondrial metabolism, but it has no link to the anti-inflammatory effects. So to do this, what we did is we used hypoxia. Hypoxia will basically induce HIF-1 alpha, and HIF-1 alpha is known to block this PDH due to inflammation, and therefore we can artificially kind of mimic a block of the TCA cycle. And we repeated the conditions, and then we measured pro-inflammatory cytokines. So what we were able to observe is that in normoxic conditions, indeed, glucocorticoids inhibit inflammation. So we measured different cytokines such as IL-1 beta, IL-6, and TNF. Surprisingly, in the absence, so during hypoxia, this anti-inflammatory effect of glucocorticoids was completely abrogated, suggesting that indeed TCA cycle functioning is important for the anti-inflammatory effects. And we also observed a similar phenomenon in human cells. So what we did is we used monocytes from healthy donors, which we differentiated into macrophages, and we performed similar experiments. And especially for IL-1 beta, we observed a very similar phenotype, suggesting that indeed the block of the TCA cycle is required for this anti-inflammatory effect. So now we could kind of link this anti-inflammatory effect of glucocorticoids with metabolic reprogramming. And we wanted to see a bit more in detail what is happening in terms of metabolism. So we performed some mass spec metabolomic analyses. And when we were looking at the glycolysis and TCA cycle metabolites that were between in pro-inflammatory macrophages treated or not with glucocorticoids, we could see that most of these metabolites were downregulated following glucocorticoid treatment, with the exception of two. So some of these examples, for example, fumarate and malate, we can see that LPS induces an accumulation of these intermediates due to basically the TCA cycle not really running. So what happens is that various metabolites will just accumulate, while glucocorticoid treatment was able to decrease their accumulation due to the TCA cycle running again. On the other hand, two of these metabolites were upregulated, so itaconate and succinate. What is well known, or has been described now quite often by Luke O'Neill's lab, who is one of the founders of macrophage immunometabolism, if we want, is that during LPS treatment, this breakdown in the TCA cycle will lead to itaconate production. And itaconate has important anti-inflammatory effects. Moreover, itaconate will block the SDH enzyme, which is responsible for converting succinate into fumarate. So there will be a massive accumulation of succinate. What we were surprised to see was that during glucocorticoid treatment, these two metabolites seem to actually be increased and not decreased. As mentioned, LPS will induce itaconate, and if we look at itaconate within the cell, we can nicely see that at early time points, there's this itaconate peak, and then itaconate production will decrease, and this is due to basically the TCA cycle just not being fed and no more production, basically no more fueling of it. However, glucocorticoids not only sustained itaconate production, but increased it. And this was noticed both intracellularly and extracellularly, suggesting that this fueling of the TCA cycle, so the glucocorticoids affecting this pyruvate dehydrogenase, pyruvate being converted to acetyl-CoA, and functional TCA cycle and mitochondria means there will be more itaconate produced. And itaconate, having important anti-inflammatory effects, could be involved in these anti-inflammatory effects of glucocorticoids. What we were able to demonstrate is when we blocked TCA cycle functioning using hypoxia, this sustained itaconate production was abolished. Indeed, indicating that functional TCA cycle is important for this itaconate production by glucocorticoids. So itaconate production is, to this date, has been described to be due to only one enzyme, IRG1. And IRG1 will convert aconitate into itaconate. And what we were expecting is that basically glucocorticoids must be affecting IRG1 expression either on mRNA level or on protein level. And what we were surprised to see is that this was not the case. And what's important to note, if I just go back quickly, is that itaconate production is only produced during inflammation. So if we treat with glucocorticoids alone, we actually don't have any itaconate. We do need the cells to be in an inflammatory state for itaconate to be produced and glucocorticoids to basically sustain this production. And this we can nicely see on the protein level. So in the absence of inflammation, we see that there is no IRG1 expression. And IRG1 is actually the most highly expressed gene during LPS treatment in macrophages. And we can see that glucocorticoids don't further induce the protein expression. And we also saw this on mRNA level, suggesting that this increased itaconate is only due to sustained functional TCA cycle and fueling of the TCA cycle, not the enzyme being affected in a certain way. And we had access to mice that do not express IRG1, isolated macrophages and treated them, and then looked at different inflammatory mediators. And what we were surprised to see is that absence of IRG1 seemed to completely abolish this anti-inflammatory effect of glucocorticoids. And a similar phenomenon was observed in humans. So there is an inhibitor of IRG1 that is available pharmaceutically. We treated the cells and we were able to observe the same. In humans, however, this effect is not as strong as in mice. And this is partially due to the fact that humans don't produce itaconate as strongly as humans. So we think that in humans, there will be other mechanisms that are also involved alongside itaconate being anti-inflammatory. And kind of the nail in the coffin to this, to demonstrate this, was going back to the first experiment, bulk RNA-seq again. And we performed bulk RNA-seq on wild-type and knockout IRG1 macrophages. And what we observed nicely is that so if you remember in the beginning, there were a lot of up and down regulated genes, but if we look at the IRG1 knockout mice, suddenly the number of down regulated genes was much less. And this anti-inflammatory effect seems to require itaconate, especially if we look at different transcription factors here on the right. We can see that in the presence of itaconate, a lot of these transcription factors are actually downregulated in the presence of glucocorticoids, while in the absence of glucocorticoids, in the absence of itaconate, this effect is not observed. So taken together, we were kind of able to piece a bit of a mechanistic approach. And what we were suggesting is that basically in LPS context, the LPS will bind to the TLR4 and lead to classical NF-kappa B activation and pro-inflammatory gene production. And this will lead first to production of itaconate and then a breakdown of the TCA cycle and reliance on glycolysis. What we're now proposing is that glucocorticoids, after binding to the receptor, somehow the glucocorticoid receptor is interacting with the mitochondria and seems to be in proximity to the PDH. And this leads to increased PDH enzymatic activity and therefore fueling of the TCA cycle and sustained itaconate production, which seems to be required for the anti-inflammatory effects of glucocorticoids in macrophages. So these were nice results, but everything was in vitro. And we then wanted to see if this could also be the case in vivo. So we utilized mouse models, and we first used an LPS-induced lung injury. So what happens is the mice receive LPS intranasal, and 24 hours afterwards, the mice were culled, and the lungs and the bronchoalveolar lavage were collected for histology and for flow cytometry and ELISA. And in the simple model using IRG1 wild type and knockout mice, we were able to observe a very strong phenotype. So if we look at the histology, we can see that during LPS treatment, there will be an infiltration of immune cells in the lung, which is quite expected. And treatment with glucocorticoids seems to reverse this phenotype. However, in the absence of itaconate in the knockout mice, this reversal was not observed. And what we were able to observe is there was actually a substantial increase in infiltrating cells, particularly of neutrophils, and that this was coupled with an increase in pro-inflammatory cytokine production, for example, TNF and IL-6. So this was very surprising because we didn't expect the phenotype to be so strong. And we went one step further because glucocorticoids are the main therapy for a wide variety of immune-mediated inflammatory diseases, including rheumatoid arthritis and asthma. And being in a rheumatology department, we really wanted to focus on rheumatology. And we were lucky enough to have access to rheumatoid arthritis patients that receive or not glucocorticoids as part of their treatment regimen. And from these patients, we collected blood and measured itaconate in the serum. And what we were surprised to see is that patients treated with glucocorticoids have higher levels of itaconate in the serum than patients who don't. And this was also observable in a mouse model of arthritis, so the KBXN arthritis model, whereby healthy mice receive serum from arthritic mice and spontaneously develop arthritis. And we can see that in these mice, glucocorticoid treatment also significantly increased serum itaconate levels. And more importantly, absence of itaconate basically inhibited the anti-inflammatory effects of glucocorticoid during arthritis. So using this KBXN model, we're able to see that wild-type mice who don't receive glucocorticoids develop clinical arthritis. Well, if we start treating the mice from day 4, where they start to have severe symptoms of arthritis, we see that mice that receive wild-type mice that receive glucocorticoids have basically a resolution of arthritis. However, in the absence of itaconate, this wasn't the case, and the mice couldn't resolve on their own. And this was also the case during asthma. So we used an ovalbumin mouse model of asthma where the mice receive ovalbumin intraperitoneal and then are basically treated again intranasal for a reactivation. And similarly to in the arthritis model, absence of itaconate abrogated the anti-inflammatory effects of glucocorticoids. So we can see histologically that that upon glucocorticoid treatment in wild-type mice, there's a nice resolution of cell infiltrate. This isn't the case in the absence of itaconate. And this is nicely observed also in flow cytometry, where absence of itaconate is linked to a massive and sustained infiltration of CD4 T cells and eosinophils, as well as to pro-inflammatory mediator production, such as IL-4, IL-5, and IL-13. So to kind of just bring this all together, we describe in the study a novel mechanism of action of glucocorticoids, where we're suggesting that the glucocorticoids after ligation to the receptor are indeed translocating to the nucleus, but also are somehow interacting with the mitochondria in order to rewire the cellular metabolism, sustaining itaconate production, and that this itaconate is key to the anti-inflammatory effects of glucocorticoids. And what we think is that this will change the way we see glucocorticoids because they are one of the most used therapies. However, one of the issues with glucocorticoids is that they do come with a lot of side effects, and many patients do have problems long term. So for a lot of therapies, the glucocorticoids can't be used long terms, or some patients even become resistant. And if we're able to bypass this genomic effect of glucocorticoids, which is for the moment associated with these metabolic side effects, maybe we're able to develop a novel class of glucocorticoid-like drugs that would have the beneficial aspects of glucocorticoids without having the side effects, and therefore maybe allow patients to be better treated and to hopefully make medicine better. So with that, I'd like to thank everyone for their attention, and happy to take questions and have a nice discussion.
Dr. Auger, thank you so much for that fantastic presentation. What interesting and groundbreaking work you've shared with us today. I'd like to open it up to our discussants to see if they would mind sharing their thoughts. Perhaps Dr. Balestrieri can share some thoughts first, and then we'll move on to Dr. Vachharajani next.
Sure. First of all, congratulations. This is truly a landmark paper. So many new findings, and if I may express my preference, the in vitro studies really point out how the mechanism that you found is applicable to type 1 and type 2 inflammation, which is usually not the case in the era of M1, M2, if anybody believes that.
Yeah.
But speaking of type 2 inflammation, I was wondering whether you think or you tested this mechanism in bone marrow macrophages activated with interleukin-4.
Yeah, so that's a good question. So we haven't tested it yet because we were really focusing on this M1 phenotype, but we would expect, or we're hoping for a similar phenotype. What we think might be different is that in, so more of an M2 IL-4 activated, the itaconate will usually not be produced because we believe that it won't be produced as much because it doesn't seem to be normally produced during M2 macrophages. So we have data from other studies where, this was, and we saw that basically itaconate didn't seem to be highly expressed just during IL-4 treatment. So we would expect there that the mechanism might be different. But we are thinking that the effect of glucocorticoids on an M2 macrophage would still involve probably metabolic reprogramming, just a different mechanism than itaconate.
And if I may ask another question? So in both in vivo models, the glucocorticoid reduce the number of myeloid cells in the lung. And because your studies focused on macrophages, which one do you think is the mechanism? There is a less recruitment, less proliferation, or these macrophages are just exhausted because the glucocorticoids seem to really drive the metabolism in the macrophages. So how long would they survive, the macrophages in the lung, after they've been exposed to glucocorticoids?
That's a good question. So in the flow cytometry, we did also look at monocyte and macrophage numbers also in the lung tissue. We didn't see any differences. So we don't think that they are dying. What we think is happening is that basically with this rewiring in the macrophages, there will just be more itaconate produced during glucocorticoid treatment, and therefore just a reduced number of chemokines, for example, by fibroblasts and endothelial cells, and therefore less infiltrating cells. But that is our current hypothesis. We haven't gone into further details.
Thank you.
Dr. Vachharajani, would you want to share some thoughts?
Sure. Well, truly a landmark paper, truly a wonderful paper. It was a pleasure reading it, especially its immunometabolism. So obviously it was great to read it.
Thank you very much.
The one question I have is, well, I study sepsis, so like I have a very skewed view of world. Everything is acute inflammation. So please forgive me if it comes across as that. But so especially so basically the bottom line, correct me if I'm wrong, that for the glucocorticoids to work, you have to have not have to have, but it's better to have working TCA cycle oxidative phosphorylation. So I'm just asking you here to speculate on a couple of things. Number one, in the sepsis world, especially when you give bugs to these cells, there is Warburg effect, and you have actually inhibition. Data suggests that there is inhibition of oxidative phosphorylation in order for the selective or aerobic glycolysis to occur. So do you think that is one of the reasons why we see such a variable effect of glucocorticoid when you give it to the sepsis patients?
I mean, that could be one of the reasons. So it's with sepsis, we also know that it's more difficult because there's this huge cytokine storm happening, and this is out of control. So maybe at that point also glucocorticoids can't rescue as much because the cytokine storm is just so huge. But it could very well be. I mean, we didn't look into a sepsis model, but I mean, hypothetically, we would expect that if the mice are not too far along in sepsis, because at one point there's just no point of return, that we would see a similar phenotype, so that in absence of itaconate and working TCA cycle, the glucocorticoids aren't working.
So a second question along the line, if I may. So to the extent that you have, say, you have to have a working TCA cycle. So what if, in other words, the PDH has to be like working? So what if you gave a DCA, like dichloroacetic acid, which inhibits the PDK, which means PDH will be working better? Are the effects similar, different? Have you done a head-to-head comparison of, say, DCA, dichloroacetic, with glucocorticoid in this area?
Yeah, so we haven't looked at that. That's something that we're interested in, because we would expect that if basically if we're inducing PDH activity, we might even be able to sustain glucocorticoid activity even more. And actually, the direction we're kind of going in is trying to see how we can sustain PDH activity without having this genomic translocation, because as I mentioned, this translocation is basically responsible for the negative side effects. So if there's a way to sustain PDH activity, that would be the best thing. So this is really the direction we're going in at the moment, and trying to also see, because we haven't evaluated really PDH activity in human cells. So this is kind of the next step, also to see if from mouse to human this is nicely translatable, or if the mechanism in humans is slightly different.
Thank you.
Fantastic. Thank you guys so much for sharing. We'll open it up for any of the audience members if you'd like to unmute yourself and ask a question, or feel free to put it in the chat and I can ask it on your behalf. Dr. Auger, I had a couple of questions. This is a really, really fascinating work. I think most of the data that you showed was a relatively acute sort of model, maybe minus the chronic arthritis patients. And as Dr. Vachharajani pointed out, sepsis is a very, very acute or maybe even hyperacute model. I'm a transplant pulmonologist by training, and we use steroids for the life of the graft and the recipient. And so my question first to you, curious if you guys have any thoughts about how this works in more chronic illness models, especially given the data that you had about the arthritis patients.
Yeah, so we don't have any data about more chronic diseases. So we are looking also at, like, we have collaborators who have chronic asthma patients, and we're trying to get samples from these patients to see, because also, especially in asthma, many patients develop basically resistance to glucocorticoids. So we're trying to see if there's in resistant patients versus non-resistant patient, and over time, since a lot of these patients take glucocorticoids for the rest of their life, to see if it's a similar phenomenon or not. So we don't have any data at the moment. That's something we're interested in though.
A couple of other questions. You guys had sort of mentioned it sounds like at least a somewhat longer term goal of this work is to identify a transcription sparing or nuclear translocation sparing sort of steroid adjacent treatment that mimics the beneficial outcomes without some of the more longer term adverse outcomes. Where are you guys in that research, and how realistic is the possibility? I mean, especially for transplant patients who are on this for life, we see osteoporosis, diabetes, I mean, all these major issues that occur. And so I'm curious as to where you guys are in that work and what you foresee that looking like. Is it really realistic that there would be something to completely sort of replace glucocorticoids, or perhaps something that could be an adjunct and reduce the amount of glucocorticoids in these sort of more long-term diseases?
Yeah, so I mean, given that the study came out so about a year now, and I'm slowly establishing myself also. So we're not that far along. What we're really hoping is we don't think that we will necessarily find a steroid that doesn't have these effects, but we're hoping to find either a small molecule or molecule that has similar effects. So that's at the stage we're at the moment, and we really do hope that longer term. So to be realistic, I don't think it will be in the next 2-3 years, but we're hoping to find a small molecule. And at the moment, we're establishing also some assays that we can basically use for screening, because if there are lots of molecules that exist and none of them have really been used to screen for these effects. So that's where we're kind of at the moment.
Very interesting. Another question. So all this amazing work was done in macrophages, and of course, macrophages are a model cell for this type of work to occur, given their ability to both be pro and anti-inflammatory, their long-lived nature, and in our world, their importance in the lung. My question is, have you guys looked at any of these? Have you done any of these experiments or looked at the role of glucocorticoids, itaconate, PDH, et cetera, in other immune cell types? As you guys may be aware, monocytes will differentiate into macrophages, but neutrophils have started to become very diverse in terms of their characterization. And we're now identifying, in the tumor literature, they're identified as myeloid-derived suppressor cells, and in other worlds, they're sort of pro-resolving neutrophils. Any data in other immune cell types on glucocorticoid effects, if this is sort of conserved, or if this is really something more towards a long-lived immune cell phenotype?
Yeah, so that's a great question, and that's actually kind of one of the directions that I'm now trying to head in with my group. So one of the things that I didn't mention, but so itaconate for the moment has been mostly described to be produced by macrophages. So dendritic cells can also produce it, and there has been one study so last year that suggests that neutrophils can also produce it, and we have some first data suggesting that eosinophils can also, but it does seem to be restricted. So T and B cells, for example, don't seem to be able to produce it, and it is only during inflammation. So in the absence of inflammation, the enzyme is not expressed at all, it seems, in any immune cells. So we do think that under the right conditions, that probably a lot of immune cells will be able to express it. And it does seem that in eosinophils, surprisingly, so we do have some preliminary data suggesting that in eosinophils, this itaconate doesn't seem to be required for the anti-inflammatory effects of glucocorticoids, for example. So in this case, we would expect another pathway, and therefore, I would also be hypothesizing that in neutrophils, there's probably also another mechanism involved, but that's something we haven't looked at. So I do know that Luke O'Neill's group, and they're very interested in itaconate, so they basically discovered it and have been, they're interested in using it also as maybe in conjunction with glucocorticoids as a therapy. And they're definitely going down that avenue, trying to see if glucocorticoids are involved in other cell types. So we're hoping this is kind of opening up a whole new world, but we also think, to be honest, that there are other mechanisms, especially given that in human cells, when we inhibit itaconate, we don't see a complete inhibition of the effects of glucocorticoids. But in human cells, it has been described more recently that fumarate, for example, also has important anti-inflammatory effects. And also succinate might be involved as well as its pro-inflammatory effects. So there might be many other metabolites that are involved. So yeah, it's a whole new field, and we're hoping that a lot of data is going to come out.
It does seem that you have opened up the floodgates to some extent here, which is fantastic. There is a question in the chat. Dr. Kurtoglu says, congratulations on the paper. I have a quick question. In mechanistic in vitro and in vivo studies, itaconate pretreatment seems to increase the tolerance to LPS-mediated injury. As a group, have you considered itaconate pretreatment in the experimental timeline if the magnitude of the response will differ when starting with ITA rather than GCs as discussed in your model?
So we have done it. So we have treated. So there are two. If you treat the cells with itaconic acid itself, it's not cell permeable, but there are two cell permeable derivatives that are on the market. And we have treated them with both. And what we see is basically just an increased effect of the glucocorticoids at that point. So this was in vitro on macrophages. We haven't done it in vivo. As I said, I know that Luke O'Neill's group is really pushing that direction, seeing as their itaconate focus. But yeah, from in vitro macrophages, at least, it seems to potentiate the effects of glucocorticoids.
Fantastic. Thank you so much for sharing. If there are no other questions in the audience, then I think that we can go ahead and conclude our webinar. So with that, I'd like to thank all the authors for participating, and in particular, Dr. Auger for the excellent presentation of his work. I'd also like to thank our discussants, Drs. Balestrieri and Vachharajani, as well as everybody who joined and submitted questions. For those ATS attendees, if there are any papers you would like presented via a webinar, please contact your assembly representatives. And with that, we will conclude our webinar. Thank you all for joining.
Thank you.
Thank you very much.